So now in translation, it is the process of protein synthesis from mRNA. Okay, it
Mhm.
occurs in the cytoplasm on ribosome in eukaryotes. Okay, so I have already told you what is mRNA. What is the full form of mRNA?
Messenger RNA.
Yes, messenger RNA. What type of masses this mRNA will carry?
The code coding for the proteins from the DNA.
Yes, yes, coding for the proteins from the DNA, but a specific protein.
Amino
Okay,
acids.
yes. Okay, so there are various steps involved in this process. See, here the transcription process completed. See, here the transcription process completed inside the nucleus. Okay, so this is first pre-mRNA. I have already told you that primary transcript. This mRNA is also called primary transcript. After few modification, this will converted into mRNA. Have you ever heard about introns and exons? Introns and
No,
exons.
no.
Okay. So during this process, some modification get happens. After that, this pre-mRNA get converted into mRNA. Okay. And
Mhm.
after these conversions, this mRNA comes on inside the cytoplasm, comes outside the nucleus and inside the cytoplasm of the cell. And as it comes outside the nucleus, what you will see that ribosome is made up of two units. Ribosome is made up of two units, smaller and bigger units. Okay, so this is the smaller unit, this is the bigger unit. Okay, so what is inside the ribosome? See what is there. See here, RNA. This ribosome has three positions. This is symbolized as E, P, and A. Okay.
Mhm.
So what you see that first of all tRNA comes at the position E. Okay.
Mhm.
First the tRNA comes. This is the tRNA. This is just like a clover leaf. Okay. This first of all at the position E, tRNA comes and get attached. This is the anticodon. If this has A, G, and U, it has the anticodon U, C, and A. Okay. After then, this tRNA shift at this position at P. This tRNA gets shifted at position P, and at this position you will see that this is already carrying a specific amino acid, and at the position P, at this position P, there is a amino acid. Okay, see, no, I have told you. See, first of all, tRNA comes at this position with the amino acid, with the amino acid. Okay, and at this position, already there is a previous tRNA. Okay, if you can call it, this is the second tRNA, so this is the first tRNA. Okay, so between the first tRNA and the second tRNA, there is a peptide bond formation took place. Okay, there is a peptide bond took place. After that, this second tRNA comes at place of first tRNA, and this is your first tRNA. This will
Mhm.
shift at this position, and ribosome will move at this position. Okay, whichever you want to say. Okay, after that, this first tRNA get detached from this position. Okay, again, if now you will see that this first position A get emptied. Again, there is third RNA, third tRNA is present. Third tRNA will come with a specific amino acid, and this amino acid again form a peptide bond with this second amino acid, and this second amino acid shift at this position, and this third amino acid gets shifted at this position, and again there is a fourth amino acid. The fourth tRNA comes with another type of amino acid, and this process will get carried on till they do not detect such type of sequence. These sequences are called stop codons.
Yep.
Okay. So if
Mhm.
ribosomes, or we can say that if ribosomes comes at this position, which is having UAA or UAG or either UGA, if ribosome detect this type of sequence, then they will stop the formation of protein. Okay. And after that, you will see that there is a growing protein chain. And when this stop codons comes, when this stop codons comes, then ribosome stops the polymerization of amino acid. Okay, and then you will see this is the first primary protein. This is called the primary protein. Okay, but I have already told you this protein is non-functional. This gene, this gene has to manufacture enzyme. This gene has to manufacture enzyme, and
Mhm.
enzyme is a type of protein. Enzyme is a type of protein. So
Mhm.
first of all, this gene encodes mRNA, and by the help of that mRNA, ribosome will polymerize protein. Okay, ribosome will polymerize protein by the help of tRNA, and this protein goes under certain modification. Then it will converted into 2-degree protein. After first modification, then it will get converted into third-degree protein, and this is the functional protein, and also this will further converted into 4-degree protein. Okay, and these third-degree and fourth-degree protein are functional proteins, or we can say that this is the enzyme. Okay, so if we summarize the steps in translation, first step is activation of amino acid. Amino acids are activated by ATP. Can you tell me what is the full form of ATP?
Uh,
Adenosine triphosphate. Okay, so this what ATP is also the energy currency of the cell. Okay,
mhm.
so first of all, by the help of ATP, amino acids are activated by the ATP and attached to their specific tRNA molecule. Okay, so initially these amino acids are hovering inside the cytoplasm, and these are inactive state. These are in inactive state. After the activation, after the activation, these amino acids get attached to the to their specific tRNA molecule. Okay, and this form a special type of complex, which which we can call it amino acyl tRNA. Okay, this is the first step of translation, activation of amino acid. Okay, So during this process, amino acids get attached to their specific tRNA. Now, the next step is initiation. Ribosome binds to the start codon. What is the start codon? AUG. What is the start codon? AUG. I have already taught you in previous session.
Mm-hmm.
Okay. So ribosome binds to the start codon, that is AUG, on mRNA. So ribosome binds to which RNA? mRNA or tRNA?
tRNA.
Come again?
tRNA.
No, ribosome attached to the mRNA. See, ribosome is attached to the mRNA. At which position? At AUG position. This is the start codon, and this codes for MET. Okay,
Mm-hmm.
understood?
Yeah.
So ribosome binds to the start codon AUG on the mRNA. Okay,
Yeah.
so the tRNA which comes first is called initiator tRNA, which will carry which amino acid? The
MET.
initiator tRNA will carry which amino acid? Methionine.
Mm-hmm.
Okay,
Yeah.
that is, you have already said it correctly. That is MET. Binds to the start codon. Okay, that is AUG. Now, the third step is elongation. Ribosomes moves along the mRNA. Ribosome moves along the ribosome moves along the mRNA, and different types of activated amino acids bring by the tRNA at specific position on the ribosome. And what is that position? The first position is A. First position is A. Okay. Now, the peptide bond forms between amino acids, and elongation process of polypeptide chains keep going. So this is the A, this is the P, and this is the exit position. This is the polymerization site, this is the exit site, this is the entry site. Okay, so between A and P position there is a formation of peptide bond between the amino acids. Okay, you should keep in your mind. Now, after the formation of polypeptide bond, there is a step, that is termination. So translation ends when the ribosome reaches a stop codon, that is UAA, UAG, or either UGA. Okay, and the polypeptide chain is released. Okay,
Mm-hmm.
and one thing you have to also keep in mind about the genetic code. These are the codes, and these are the genetic codes, that is triplet. These are the genetic codes. Okay, so the genetic codes are always triplet. Three nucleotide bases encodes one amino acid. Okay, so we always say that genetic codes are triplet, and genetic codes are always universal. Either you will study in eukaryotes or prokaryotes, animals, or in microbes, in fungi, or in aquatic animals, any type of organism, the genetic code is always universal. If genetic code for AUG is responsible for methionine amino acid, then this will never change. But there is exception for methionine in the mitochondria. Okay. So do you know that mitochondria has the capacity to do translation? Do you know that?
No.
Okay. So translations also occurs inside the mitochondria. Okay. We
Mhm.
will see that in our sessions.